everyone, this is Ms. Snook, your science teacher, and today I'm going over lab lesson 3.13, which is the Human Digestion lab part 1. In 2 days, you'll be doing the lab Human Digestion part 2, which is 3.15. This is a 2-part lab. Today is getting the lab set up, getting some information to fill out on your student guide and on your lab report, and then in 2 days we'll come back and check the results of our lab. Lab 3.13 can be accessed in the LMS on the left-hand side under Unit 3. Click on the link to access the lab. This is what the page looks like, and when you click through here, you'll be able to read the Explorer and then come to the Human Digestion Virtual Laboratory. You will only need your computer for this lab. The lab report that you will need is up in Doc Sharing. When you click on Doc Sharing, it will be in the left-hand side. It'll say Lab 3.15 Doc. That is the lab report you're using for 3.13 and 3.15. It's one lab report for both parts of this lab. You will also need to open the student guide, which has the instructions for the lab, which is located on page 4, which is after the link for the virtual lab. Go ahead and click on the virtual lab link. It's going to take you to a site outside of the LMS, to a page that looks like this, and you will notice there's a tutorial, then a begin lab button, and then an enter code. This enter code button is where you will be entering a code that we're going to get at the end of today in 2 days when you come back to view your results. I recommend you do the tutorial so you can practice manipulating the virtual lab, and then when you're done, come back and begin the lab. Go ahead and choose your glove colors, and we're going to go to Part B in the student guide, which is the milk digestion part of this lab. The Part A is doing the tutorial. Okay, so Part B, we're going to be working with this set of test tubes right here. You can zoom in on these to see what they say. Oops, I accidentally picked one up. And this is the milk test that we're doing. So we're going to go ahead and grab our pipette and fill it up with some milk. Come down and put it in each one of our test tubes. Oops. And I'll go ahead and put the pipette down. And then we're going to, in test tube 1, we're going to add some water. And we need 2.5 milliliters of water, which is 1 squirt of our pipette into the graduated cylinder. Go ahead and pick up our graduated cylinder, and you have to get it right there in order to tip it in. Next, we're going to add hydrochloric acid to our second test tube, and then we're going to add lactase to our third test tube, and then we're going to add lactase and hydrochloric acid to our fourth test tube. Next, we're going to grab some stirring rods, 1 each for each test tube, and stir our solution. Next, we are going to put stoppers on our test tubes. It gets a little funky. You gotta get it just right there. The hand opens. That's how you know you have it. There you go. All right. And the student guide it explains to you what's going to happen when milk gets digested in the presence of lactose. So go ahead and read that. And we're going to have to test the milk test tubes with these test strips that are up here 2 days from now to see if there's glucose present in our test tubes. But we're not going to do that today. The next thing you're going to do is go to your lab report and write down your predictions So you're going to open your lab report. Remember, this is the one that comes from doc sharing, not the one that's at the end of the lab. Please do not use that. You'll fill out your hypothesis, but the hypothesis is for the second part of the lab, which we're going to get to in just a moment. So we're not filling that out yet. Number 1 is also for the second part of the lab. So you're going to skip on down here to this chart where you have your milk digestion data. What you're going to do is fill out your prediction in column 1. And then in 2 days we'll come back, test with our glucose strips, and answer this question. Next, we're going to go to part 3 of our lab, which is the protein digestion, and we're going to use an egg. The egg is to the right of these 6 test tubes right here on the counter, and it says to mince the egg, so you just grab it and it cuts it up for you. Isn't that amazing? And then we're going to grab our spoon, put some egg in each one of our test tubes. And go ahead and put our spoon down. And the first test tube, we're going to put 10 milliliters of water. So we're going to come back over here, grab our water pipette, go up here, and since this is a 2.5 milliliter pipette, we're going to need 4 squirts to get it up to 10. Go ahead and grab our graduated cylinder and come over here and pour it in our test tube. All right, next we're going to add 10 milliliters of pepsin. So we're going to come over here, grab our pepsin. This is also a 2.5 milliliter dropper, so we're going to need 4 squirts of it. And one more. And put our lid back on. Go ahead and grab our graduated cylinder, come over and pour that into our test tube. Now we're going to come over and rinse our graduated cylinder out and put it down. The next test tube we're going to make is our acid, and you can zoom in to make sure you're putting the correct chemical into each of these by pressing on this button down here. But if you're following along in the student guide, it goes in order. So we're going to go ahead and grab 10 milliliters of our acid. And pour that into our third test tube. All right. Oh, and now we're going to go rinse that out at our sink. Good. The next test tube we're going to make is going to be the one with the base, and we're using sodium bicarbonate as our base. Oops. And we'll go ahead and grab that, bring it over to our test tube. Pour that in. I'm going to just put my graduated cylinder right. Oops, I need to rinse it out, sorry. I'm going to rinse it out over here. Come back over here. I'm just going to put it closer to where we're working. Next, we're going to do pepsin again. And we're going to do 5 milliliters of the pepsin, which is 2 squirts of the dropper. 2.5 times 2 is 5. And then we need 5 milliliters of the base. So 2 squirts of that. And we get a total of 10 again. Come over here and pour that into our 4th test tube, or 5th test tube actually. Go ahead and rinse that out. And we're going to come right here. And last but not least, we have our pepsin and acid. So we're going to grab 5 milliliters of pepsin, 2 squirts worth. And then our acid is the hydrochloric acid, 2 squirts of that. And we'll go ahead and put that in right here. Great. I'll go ahead and rinse out my graduated cylinder. Want to keep my lab area neat. Put that down. And then I'm going to grab my stirring rods, stir each one of these. Make sure you use a clean stirring rod for each test tube. I like the sound effects. It sounds like you're actually stirring in a glass test tube. It's kind of cool. Alright, and last but not least. Alright, and then now for this one, we actually have to go over and grab a test strip. We're going to grab our forceps, and we're going to grab a pH paper and come over here and test each one of these tubes. That looks dirty. Why did it turn green? It shouldn't turn green. We got to put that in the trash can. Drop, drop, drop. Where's the trash? Sometimes they have a trash thing on here. So I got to get this just right to test. And this is where the lab gets a little tricky. Let's see if I can grab another test strip. I can't seem to put that in the trash. I can't get rid of it. This is terrible. Oh, it looks like I want to drop it on the floor here. Oh, goodness. Bloopers. This should go on the virtual lab bloopers, huh? I can't let go of this test strip. It's stuck to me forever.